Hello everyone, welcome back again to Chinmoye's Biology channel. So today we are here back with a new episode of Dihybrid cross. So till now in Mendel's inheritance we have studied about the monohybrid cross, codominance, incomplete dominance, about multiple alleles, single alleles and all. So today it's a bit different topic and we are going to study about inheritance of two genes. So this is very important lesson for your 12th CBSE biology as well as ICSE also. So let's get started without further delay. So what are the today's learning objectives? So today we are going to study about the inheritance of two genes and that is the dihybrid cross. We are going to study and about the law of independent assortment. So without any further delay, we will start with these two important topics and please don't skip the lesson in between. Keep watching the full lesson because you will be getting many important knowledgeable things from these two topics which I am going to teach you today in this lesson. So let's get started. So here we are going to start about the inheritance of two genes, right? So Mendel also worked with and crossed pea plants with two different characters. So we are talking about two genes here. Till now in monohybrid cross we have crossed with a single character. So now we are crossing the pea plants which are having two different characters, as is seen in the cross between a pea plant that has seeds with yellow color and round shape and seeds with green color and wrinkled shape. So two characters, yellow color and round shape. And we are seeing here green color and wrinkled shape. Okay, so green color we will write as small y and wrinkle we will write as small r. Okay, so we will not write G and W. Okay, so we are going to represent this as small y and small r. Okay, and this one is capital R and capital Y. So these are dominant characters and these are recessive characters. Now let us see, Mendel found that the seeds resulting from the crossing of the parents had yellow colored and round shaped seeds. Now here can you tell which of the characters in the pair yellow and green color and round and wrinkled was dominant? Now how we are going to say? Now as we proceed further we will be able to say that which one is dominant and which one is recessive. Now yellow color was dominant over green and round shape was dominant over wrinkle. So for that only I have written this Y as capital and this R as capital. We know that yellow, the things we are representing as dominant, we are writing them in the capital letters. Okay, and the recessive ones are written in small letters. So yellow color was dominant over this green color and round shape was dominant over this wrinkle shape. Now these results were identical to those he got when he made separate monohybrid crosses between yellow and green seeded plants and between round and wrinkled seeded plants. So round and wrinkled seeded plants and yellow and green seeded plants, they were crossed. Now let us see the genotypic symbols. So this Y is for dominant yellow seed color, Y is for recessive green seed color, R is for round shaped seeds and this small r is for wrinkled shaped seed. So this I have told you earlier. Now the cross between the genotype of the parents can then be written as how you are writing the genotype of the parents. RR is round and YY is yellow. So round yellow seed is having the genotype of RR and YY. And round and wrinkled and green seed is having a genotype of small RR and small YY. Is it clear to everyone? So capital RR and capital YY is the genotype of the parent having round seed and yellow color, and small RR and small YY is the genotype of the seed having wrinkled and green color. Okay, now the cross between the two parents can be written down as shown in figure 4.7. So we will see down the figure, the Punnett square, how it is written and how it is shown at the genotypes as well as the phenotypes. Okay, so the gametes RR and RY, they unite on fertilization to produce the F1 hybrid plant, that is RR and YY. Okay, so RY and RY, they are being crossed, they are being fertilized to produce the F1 hybrid of RR and RY. Okay, so this RY and RY, when they are crossed, they are giving you the F1 hybrid of RRYY. So is it clear? So they are giving you a F1 hybrid of RRYY. So when the Mendel self-hybridized the F1 plants, he found 3/4 of F2 plants had yellow seeds and 1/4 has green seeds. So what is the meaning of self-fertilizing? We know that this is again crossed with the same type of genotype. So this is self-fertilization. So then he is getting what? He is getting a ratio of 3:1. Okay, so that is what? That 3 are yellow seeds and 1 are green seeds. Okay, now the yellow and green colors segregated in the ratio 3:1, and round and wrinkle seed shape also segregated as 3:1. So this is about the yellow and green seeds. And what about this round and wrinkled? Sorry. So round and wrinkled also, they came as 3:1. So round and wrinkled also, they came as 3:1. So round is 3 and wrinkled is 1. So this is the wrinkled and round ratio. So this is what came. So it is just like the monohybrid cross. So let us move further when it was again crossed. So what happened in the plants? Let us see in the F2 generation. Okay. So here let us see the parental generation or the P generation here. Here the genotypes we have told, round and yellow is RRYY, and round and wrinkled is, wrinkled and green is RR, small r, small YY. So the gametes are RY from here and RY from this wrinkled and green seed. Now what is happening when in the F1 generation this RR and YY, from this we are selfing. So we are doing a self cross. We have studied it earlier in the main monohybrid cross as well. So these are the female gametes and these are the male gametes. So this is RY, RY, RY and RY. Like this we are writing in the left side of the Punnett square as we were writing in the monohybrid cross, like that only. As we are self-crossing, what does that mean? That we are doing a cross between this and again the same alleles will be there. So how we are writing that? This will be written in the left side as a female part and this will be written as the male gamete. So these are the alleles. Okay, so let us do the Punnett square, complete the Punnett square. So the first one is RR, RY, RR, YY, it will come. The next one is RR, RY. Okay, so the next one is RR, Y, capital Y, small y. So you know how to write, right? This one, this one, this one, this one, this one, this one. You are supposed to multiply this to complete the Punnett square. So you are taking this one and first you are multiplying it with all this 4. Next you are taking this and again multiplying it with all this 4. Then again this and again multiplying this with all this 4. And again this last one and you are multiplying this with all 4. So then we are getting this result, right? So what we are seeing? We are seeing visibly that there are 9, 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 round and yellow colored seeds. Okay, 1, 2, 3. 3 yellow but wrinkle seeds. 1, 2, 3. 3 round but green color seeds. And this last one is green as well as wrinkle seeds. So what phenotypic ratio we are getting? 9:3:3:1. Is it clear? So, in this F2 generation, we are getting the phenotypic ratio of 9:3:3:1. Is it clear to all of you, right? It should be clear. You just write this Punnett square with your own, like in your answer book, you make the squares. 16 squares you make out, and you write the female gametes here, the male gametes here. You just cross it. So once you cross it, you will see what results you are getting, and the ratio will come as 9:3:3:1 phenotype. So let us move on down. So round and yellow 9, round and green 3, wrinkled and yellow 3, wrinkled and green 1. Okay, the results of dihybrid cross where the two parents differed in two pairs of contrasting traits, seed color and seed shape. You are giving this as a phenotype. Now we will move on to the law of independent assortment. So what is it? So we are going to discuss about the law of independent assortment. So what is that? What is law of independent assortment? So in the dihybrid cross, we have discussed about the phenotypes, that is the phenotypic ratio is 9:3:3:1. We have discussed that round and yellow, wrinkled and yellow, round and green and wrinkled and green, they are appearing in the ratio of 9:3:3:1. So that we have already done as per the Punnett square. Okay, so such a ratio was observed by Mendel by taking different other characters as well. Okay, so now we can say that in several other pairs also it has been done. So the ratio of 9:3:3:1 can be derived as a combination series, like 3 yellow is to 1 green, 3 round is to 1 wrinkled. Okay, so all the characters are here independently found. Okay, now this derivation can also be written as 3 round is to 1 wrinkled, 3 yellow is to 1 green is equal to 9 round and yellow is to 3 wrinkled and yellow is to 3 round and green is to 1 green and wrinkled. So just a different combination you are using here. Now, based upon such observations on dihybrid crosses between plants differing in two traits, Mendel proposed a second set of generalization that we call what? Laws of independent assortment. And this is a very important law. So according to this law, what he told that when two pairs of traits, like here we have taken round and wrinkled, here we have taken round as well as wrinkled, and we have taken yellow as well as green. So two pairs of traits are combined in a hybrid, then segregation of one pair of characters is independent of the other pairs of characters. So segregation or separation of one pair of characters is independent of the other pair of characters. How? So we are using the Punnett square effectively to understand this independent segregation of two pairs of genes during meiosis. What is that? During the production of eggs and pollen in the F1 generation. So the plant is RR and YY. So consider the segregation of one pair of genes as R and another pair as R. So the eggs and pollens, you need to find out how the genes are segregated. How? So one pair is R and R, one pair of genes. So the other pair should be Y and Y. Okay. So how it is? 50% of the gametes have the gene R, whereas the other 50% have R, small r. Now besides each R and R, it should also have the alleles Y and Y, that is yellow and green. Okay, this was round and wrinkled. Now the main important thing is what to remember, that segregation of 50% of R and 50% of R, small r is independent from segregation of 50% of Y, that is capital Y yellow, and 50% of small y. Therefore, 50% of R bearing gametes has Y and 50% has small y. The same thing happens, that is 50% of the R bearing gametes are having this Y and other 50% is having this Y. So This small r is also having y and y, capital Y and small y. Capital R is also having small y and capital Y. So the 4 genotypes of gametes, that is of 4 pollen and 4 eggs, will be RY, capital R, capital Y, capital R, small r, capital Y, and small r, small y. So these are the 4 types of pollen and egg. Okay, so these are the gametes. Now each with a frequency of 25% or 1/4 of the total gametes produced. Okay, so when you write down the 4 types of eggs and pollen on 2 sides of a Punnett square, you know how we are getting the answers, right? So we know how to write in the Punnett square. We are multiplying the side of the female with that of the male, and we are getting the Punnett square box. Okay, so it is very easy to derive the composition of zygotes from these gametes, one written in the left side and one on the right side. So they will give rise to the plants of F2 generation, right? So although there are 16 squares, how many different types of genotypes and phenotypes are formed? So that we know how it is found out. There are not 16 types of genotypes, or there are not 16 types of phenotypes. So there is a ratio of phenotypes and genotypes, though there are 16 squares. Note them down in your book or copy. Now, here the genotypes found in the F2 generation, we will discuss about them, and phenotypes we already know they are 9:3, 3:1, and so that all in details we will be discussing in the next 2 slides. So let's move on to those slides. So here we will do again a quick revision of the phenotypic ratio, though we have already studied it. So let us see that. So let us find out that round and yellow phenotypes. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So these all are round and yellow phenotypes. So that is, there are 9 in number. Next comes round and green phenotype. That is 1, 2, 3, right? So round and green phenotype are 3 in number, so 3. Next comes wrinkled and yellow, that is 1, 2 and 3 in number, so it is 3. And wrinkled and green is 1 in number, so it is 1. So then it is 9:3:3:1. So this is the phenotypic ratio of the dihybrid cross, which we have discussed earlier. Now we will move on to the genotypic ratio. So phenotypic ratio we have done already, and this is the genotypic ratio. So this is a bit difficult, but we will find it out and sort it out. So the phenotypic ratio is 9:3:3:1. So the genotype will be like this. You can find out that YY and RR is only 1. So this type, that is this genetic type, genotype is only 1. YY, R, capital R, small r are 2. So capital Y, capital R, small r are only 2. This one and this one. So this is only 2. So here we can write 2. Then capital Y, small y, and both capital R are again are 2. So this are again, you can find out, this are again 2. So again 2. Then capital Y, small y, and capital R, small r are 4 in number. Okay, so 1, 2, 3, and 4. Okay, so this are 4 in number. So you can write there 4. Okay, then capital Y, and small r, small r. So here let us find out only 1 is there. Okay, now small y, and capital R is 1 in number. Sorry, this capital Y, small y, and small r is another one is there. Okay, so these are 2 in number. So genotypic ratio is a bit difficult, so you need to search out properly. So this capital Y, and small r are 2 in number. This one, and we have seen this one. So 2 in number. Okay. Now next comes small y and capital R. This is only 1 in number. Next small y and capital R, small r. This is here 1 and here 1. So 2 in number. So here we can write the genotypic ratio. Here once again 2 we can write, and last one is YY and RR. Both in small number, they are only 1. So the ratio full and final will be a big one, a vast one, that is 1 is to 2 is to 2 is to 4 is to 1 is to 2 is to 1 is to 2 is to 1. So this is the genotypic ratio and phenotypic ratio. We have revised many times. We all know that. So please do go through this full episode. Don't skip it in between, and let's wait for the next part. And what will be the next part? That is, we will read regarding the chromosomal laws of inheritance. Okay, so that is very important, and so stay tuned, stay updated, so that whenever I am posting the episodes, you will get a pop-up. So don't forget to share your comments, share your views. If you are having any doubts, any queries, if you want me to describe or discuss any lesson, please do write in my comment box so that I can get back to you with that lesson. Okay, so stay tuned, and thanks a lot for listening and watching to my videos. Okay, stay updated with all the lessons. Thank you. Thanks a lot for watching.